Biotechnology in medicine. Scope of this topic. This topic explores how modern biotechnology is applied in medicine, from molecular foundations to clinical therapies. We will examine biological principles behind biotechnology, core molecular techniques, medical applications and case examples, ethical, regulatory, and future perspectives. Defining biotechnology. Biotechnology is the application of biological systems, organisms, or derivatives to develop products and technologies for human benefit. Medical biotechnology specifically focuses on disease diagnosis, disease treatment, disease prevention, regenerative and personalized medicine. At its core, medical biotechnology integrates biology, chemistry, engineering, and medicine. Historical context. Early medicine relied on natural extracts, empirical treatments, limited understanding of disease mechanisms. The biotechnology revolution began when scientists learned to manipulate DNA, control gene expression, produce human biomolecules artificially. This shift moved medicine from symptom-based treatment to mechanism-based intervention. Why biotechnology changed medicine? Traditional pharmacology often targets biochemical pathways broadly, which can cause off-target effects. Biotechnology allows molecular precision, target-specific therapies, human-identical biological drugs. This precision is essential for treating genetic diseases, cancer, autoimmune disorders, rare and orphan diseases. Slide 6. Cellular basis of biotechnology. All biotechnological processes depend on cells. Cells function as gene expression systems, protein production factories, self-replicating units. Understanding cell biology is essential because biotechnology exploits normal cellular processes for therapeutic purposes. Types of cells used in medical biotechnology. Different host systems are selected based on protein complexity. Prokaryotic systems, for example bacteria, high growth rate, simple genetics, limited post-translational modification. Eukaryotic systems, yeast, mammalian cells, complex protein folding, glycosylation capability, required for antibodies and vaccines. DNA as the molecular foundation. DNA stores biological information in nucleotide sequences. In medicine, DNA analysis allows identification of disease-causing mutations, design of gene therapies, development of molecular diagnostics. Medical biotechnology fundamentally involves reading, copying, modifying, or delivering DNA. Genes, expression, and disease. Genes encode proteins via transcription, DNA to RNA, translation, RNA to protein. Disease can result from mutated genes, overexpression or underexpression, improper protein folding. Biotechnology intervenes at any stage of gene expression. Proteins as therapeutic molecules. Proteins perform nearly all biological functions. Therapeutic proteins include hormones, insulin, enzymes, asparaginase, growth factors, antibodies. Unlike small molecule drugs, proteins are highly specific, structurally complex, biologically active. Enzymes in biotechnology. Enzymes act as biological catalysts. Key enzymes in medical biotechnology: restriction endonucleases, DNA ligase, DNA polymerase, reverse transcriptase. These enzymes enable DNA manipulation with molecular precision. Introduction to recombinant DNA technology. Recombinant DNA technology involves combining genetic material from different biological sources into a single DNA molecule. This allows expression of human genes in non-human hosts, large-scale protein production, standardized pharmaceutical manufacturing. Molecular steps. Gene isolation. The target gene is identified through genomic analysis, amplified using PCR, verified by sequencing. Accuracy at this stage is critical, as even a single nucleotide error can alter protein function. Restriction enzyme digestion. Restriction enzymes recognize specific DNA sequences, create sticky or blunt ends, enable precise gene excision. This specificity ensures controlled DNA recombination. Vectors and plasmid design. Vectors are DNA carriers. An effective plasmid vector contains origin of replication, selectable marker genes, promoter sequences, multiple cloning sites. Vector design determines expression efficiency and protein yield. Transformation and transfection. DNA delivery into host cells occurs via transformation, bacteria, transfection, eukaryotic cells, viral vectors, gene therapy. Each method balances efficiency, safety, and stability. Expression system optimization. Gene expression depends on promoter strength, codon optimization, host cell physiology. Graduate-level biotechnology focuses heavily on expression optimization to maximize yield and stability. Bioreactors and scale-up. Biopharmaceuticals are produced in bioreactors. Controlled parameters include temperature, pH, oxygen, nutrient supply. Scaling up from lab to industry is one of the biggest challenges in biotechnology. Downstream processing. After expression, proteins must be isolated, purified, stabilized. Techniques include chromatography, filtration, ultracentrifugation. Purity directly affects safety and regulatory approval. Biopharmaceutical drugs. Biopharmaceuticals are large, complex molecules produced in living systems, highly sensitive to conditions. Examples: insulin, growth hormone, interferons, erythropoietin. Monoclonal antibodies. Molecular basis. Monoclonal antibodies bind specific epitopes on antigens. They exploit immune specificity, antigen-antibody binding, immune effector mechanisms. Production of monoclonal antibodies. Traditional method: hybridoma technology. Modern methods: recombinant antibody engineering, humanized antibodies, fully human antibodies. Graduate students should understand antibody engineering strategies. Mechanisms of action in cancer. Monoclonal antibodies block growth factor receptors, induce immune-mediated cell death, deliver cytotoxic payloads. This allows targeted cancer therapy with reduced systemic toxicity. Genetic engineering in medicine. Genetic engineering modifies DNA to study gene function, correct mutations, introduce therapeutic genes. It underpins gene therapy and synthetic biology. Gene therapy. Conceptual framework. Gene therapy treats disease by adding functional genes, silencing defective genes, editing genomic sequences. The goal is long-term or permanent correction. Gene delivery systems. Delivery vectors include viral vectors, AAV, lentivirus, non-viral vectors, liposomes, nanoparticles. Vector choice affects efficiency, safety, immune response. Clinical applications of gene therapy. Used for inherited disorders, certain cancers, immunodeficiencies. Challenges include immune reactions, insertional mutagenesis, cost. Stem cell biology. Stem cells are defined by self-renewal capacity, differentiation potential. They form the basis of regenerative medicine. Types of stem cells. Embryonic stem cells, pluripotent. Adult stem cells, multipotent. Induced pluripotent stem cells, reprogrammed. Each type has distinct ethical, technical, and clinical implications. Regenerative medicine. Regenerative medicine aims to repair damaged tissues, restore organ function, replace lost cells. This represents a shift from treatment to biological restoration. Molecular diagnostics. Biotechnology enables diagnosis at DNA level, RNA level, and protein level. Early molecular diagnosis improves prognosis and treatment outcomes. PCR. Principle and medical use. PCR amplifies DNA exponentially using DNA polymerase, primers, thermal cycling. Applications: infection detection, genetic screening, cancer diagnostics. Biotechnology in vaccine development. Modern vaccines use recombinant proteins, genetic material, mRNA, viral vectors. These approaches improve speed, safety, scalability. Ethical and regulatory considerations. Ethical issues include genetic privacy, human enhancement, access inequality. Regulation ensures safety, efficacy, ethical compliance. Future directions. Emerging trends. Personalized medicine, gene editing technologies, AI-guided drug discovery, precision diagnostics. Final synthesis. Biotechnology in medicine integrates biology with clinical practice, enables precise, targeted therapies, raises ethical responsibilities, defines the future of healthcare.
Transition from medical to industrial biotechnology. While medical biotechnology focuses on patient-centered outcomes, industrial biotechnology focuses on large-scale production, cost efficiency, sustainability, process optimization. Despite these differences, both fields rely on the same molecular and cellular foundations. What is industrial biotechnology? Industrial biotechnology is the application of biological systems, microorganisms, enzymes, and cells to produce goods and services for industry. It is often referred to as white biotechnology, distinguishing it from red biotechnology, medical, green biotechnology, agricultural. Industrial biotechnology integrates molecular biology, biochemical engineering, systems biology, and process control. Its primary goal is to replace or improve traditional industrial processes using biological efficiency. Why industry turned to biotechnology? Traditional industrial processes often rely on high temperatures, high pressures, toxic chemicals, and non-renewable resources. Biological systems, in contrast, operate at mild conditions, are highly specific, use renewable raw materials, and produce fewer harmful byproducts. This shift supports economic efficiency, environmental sustainability, and regulatory compliance. Core principle: biological catalysis. At the heart of industrial biotechnology is biological catalysis, primarily through enzymes and living cells. Biological catalysts accelerate reactions dramatically, reduce energy requirements, and increase product specificity. Unlike chemical catalysts, enzymes are highly selective, biodegradable, and adaptable through genetic engineering. This catalytic advantage underpins all industrial bioprocesses. Role of microorganisms in industry. Microorganisms are the primary workhorses of industrial biotechnology. Commonly used groups include bacteria, yeasts, and filamentous fungi. They are selected because they grow rapidly, can be genetically modified, and perform complex biochemical reactions. Industrial strains are often highly engineered, far removed from their natural counterparts. Strain selection and strain improvement. Industrial success depends heavily on strain performance. Key characteristics of industrial strains: high productivity, genetic stability, resistance to stress, and ability to utilize cheap substrates. Strain improvement techniques include classical mutagenesis, adaptive laboratory evolution, recombinant DNA technology. At the graduate level, strain optimization is viewed as a systems-level challenge. Genetic engineering for industrial strains. Genetic engineering allows precise modification of microbial metabolism. Typical objectives include overexpression of key enzymes, removal of competing pathways, improved substrate uptake, increased tolerance to products. These modifications redirect cellular resources toward maximum product yield, a concept known as metabolic flux optimization. Metabolic engineering, core concept. Metabolic engineering is the deliberate modification of cellular metabolic pathways to increase production of a desired compound. It involves pathway analysis, flux balance modeling, gene regulation control. Unlike single gene manipulation, metabolic engineering treats the cell as an integrated biochemical network. Systems biology in industry. Systems biology complements metabolic engineering by providing genome-wide data, transcriptomic analysis, proteomic profiling, and metabolomic measurements. These data allow scientists to understand global cellular responses rather than isolated reactions, improving predictability and scalability. Industrial fermentation overview. Most industrial biotechnology processes rely on fermentation, which is controlled microbial growth for product formation. Fermentation is not limited to alcohol production. It is a general bioprocessing platform. Industrial fermentation involves cell cultivation, substrate conversion, and product accumulation. Types of fermentation processes. Major fermentation modes include batch fermentation, simple, flexible, widely used; fed batch fermentation, controlled feeding to prevent inhibition; continuous fermentation, steady state operation, high productivity. The choice depends on product type, organism, and economic constraints. Bioreactors in industrial biotechnology. Bioreactors are engineered systems designed to support biological reactions. Key design considerations: mixing efficiency, oxygen transfer, temperature control, sterility. Bioreactor design directly affects cell growth, product yield, and process reproducibility. Scale-up challenges. Scaling from laboratory to industrial scale is one of the most difficult aspects of biotechnology. Challenges include oxygen limitations, shear stress, heat removal, nutrient gradients. Processes that work at 1 liter may fail at 100,000 liters without careful engineering. Upstream processing. Upstream processing includes all steps before product recovery, such as media preparation, sterilization, inoculum development, fermentation control. Optimization at this stage determines the upper limit of productivity. Downstream processing. Downstream processing refers to product recovery and purification. This often represents 50 to 80% of total production cost. Steps include cell separation, product extraction, purification, formulation. Efficient downstream design is critical for commercial viability. Enzymes as industrial products. Enzymes themselves are major industrial products. Industries using enzymes include detergents, food processing, textiles, paper, and pulp. Enzymes replace harsh chemicals while improving process efficiency. Enzyme engineering. Natural enzymes are often not optimal for industrial conditions. Protein engineering techniques include directed evolution, rational design, computational modeling. Goals include higher stability, broader pH tolerance, improved activity. Biotechnology in the food industry. Industrial biotechnology plays a central role in food production. Applications include fermented foods, flavor enhancement, nutritional improvement, food preservation. These processes rely on controlled microbial metabolism. Bio-based chemicals and materials. Industrial biotechnology enables production of organic acids, alcohols, bioplastics, biosolvents. These products reduce dependence on petroleum-based chemistry. Biofuels and energy. Biotechnology, microorganisms can convert biomass into bioethanol, biodiesel, biogas. Energy biotechnology integrates microbial metabolism, process engineering, sustainability analysis. Biotechnology and sustainability. Industrial biotechnology supports circular economy, waste valorization, carbon footprint reduction. Biological processes transform waste into valuable products. Environmental biotechnology. Microorganisms are used for wastewater treatment, bioremediation, pollution control. These processes exploit natural microbial pathways to detoxify environments. Regulatory and safety aspects. Industrial biotechnology must comply with biosafety regulations, environmental protection laws, quality standards. Containment, monitoring, and risk assessment are essential components of industrial operations. Economic considerations. Commercial success depends on cost of raw materials, process efficiency, market demand, scalability. Economic analysis is as important as biological innovation. Future of industrial biotechnology. Future directions include synthetic biology, AI-driven strain design, fully automated bioprocessing, carbon-negative production systems. Industry is moving toward digitally optimized biological factories. Integration with medical biotechnology. Industrial biotechnology provides production platforms for medical products, scalable manufacturing systems, cost-effective biopharmaceutical supply. Thus, industrial and medical biotechnology are deeply interconnected. Final synthesis. Industrial biotechnology applies biological systems at scale, replaces chemical-intensive processes, supports sustainability and innovation, bridges science, engineering, and economics. It represents a foundational pillar of the modern bioeconomy.